Our body is a nutrient-rich, warm and moist environment which remains at a uniform temperature and constantly renews itself. For these reasons, the human body provides a suitable environment for microbial invasion. Fortunately, we have a wonderful immune system that consists of several lines of defense against such threats. The first line of defense is the barrier defenses, which protect us from the enormous amount of pathogens that are trying to invade our body's hospitable environment. This line of defense is represented by all our exposed surface epithelia, such as that of the skin, the respiratory tract, the gastrointestinal tract, and the urogenital tract, which altogether provide a critical barrier to the outside world. And a lot of pathogens actually are stopped by this barrier alone, without the need of additional assistance. Now, if the first line's barrier integrity is breached and the pathogens advance forward into our body's interior, then our second line of defense will sense the presence of these pathogens and will come into play, making sure these pathogens will not go any further. This second line of defense is provided by an array of cells that, when induced, can trigger a variety of cellular responses. These two lines deal with the vast majority of infectious agents, and only a relatively small amount of pathogens are able to withstand and breach these two lines. Now, if a pathogen breaches the second line and have a high antigen threshold that overwhelms our second line defense mechanisms, then our second line will call for help and will result in the activation and recruitment of our third line adaptive defenses. This third line of defense is provided by antigen-specific lymphocytes that result in a state of protective immunity. Now, in this video, we will discuss these three lines of defense and how they shape our immune response. But before getting started, I want to ask you kindly to support us and help us make more videos by hitting the like button, leaving a comment down below, sharing it with your friends, and please don't forget to subscribe to our channel. Now let's get started. The first line of defense is provided by a set of barriers, namely physical barriers, chemical barriers, biological barriers, and also mechanical defenses. These barrier defenses are continuously present and ready for action and form the interface between the environment and the body. Now, if this line of barriers is breached, then the second line induced defenses comes into play, and these induced cellular defenses are executed by our innate immune cells that are either present at or underneath the breached epithelium or recruited as a result of that breach. And if the invading pathogen reaches a certain antigen threshold, then the second line defenses will activate the third line adaptive defenses, which consist of various antigen-specific lymphocytes and their products. The first two lines of defense are considered part of innate immunity, whereas the third line of defense is considered part of adaptive immunity. Now let's take a closer look at our first line of defense. The first of these barrier defenses are the physical barriers. The physical barriers consist mainly of the epithelial cells of various tissues that are joined together by tight junctions. Each epithelial surface has its unique defense strategies against the microbes they typically encounter. This epithelia may be thick and formed of multiple layers of cells, such as that of the skin, which has an outer part called the stratum corneum, and that alone consists of about 15 layers of flattened, impermeable dead cells that gives skin its strength. The epithelia could also be thin and formed of a single cell layer, such as that of the colon, which has an outer part formed of 2 layers of thick mucus. This mucus establishes a viscous physical barrier that impedes microbial access to the epithelial surface below it. The second of our first-line barrier defenses are the chemical barriers. The chemical barriers provide innate immune protection against infection by chemical means. And an example on that are the antimicrobial proteins and peptides, the complement proteins, and the acidic pH. The antimicrobial proteins and peptides are present in all body fluids and act as a natural antibiotic that can kill many pathogens or weaken their effect. The complement proteins are present in serum and interstitial tissues and can initiate a local inflammation at the area of invasion, can enhance pathogen killing by our innate immune cells, and can even kill pathogens directly by putting holes in their membranes. The low pH of many surfaces provide an acidic environment that either kills pathogens or inhibits their growth. Now, the next of our first-line barriers is the biological barrier, which is provided by our commensal microbiota that is normally resident at barrier tissues. The microbiota represent the non-pathogenic microorganisms that normally live on our body's epithelial surfaces harmlessly in a symbiotic relationship. This friendly microbiota act as a barrier against pathogenic microorganisms. Pathogens have to compete with our normal microbiota for nutrients and areas of attachment, which makes it difficult for their survival. Additionally, the normal microbiota can actually kill pathogens directly. In addition to the physical, chemical, and the biological barriers, the first-line defenses also include mechanical mechanisms of pathogen elimination, such as coughing, sneezing, urination, and defecation, all of which expel pathogens outside of the body. These barriers altogether form our innate immune system's first line of defense against infection. Now, despite the strong physical, chemical, and biological barriers of this line of defense, some pathogens may be able to disrupt and breach this line and pass through its solid defenses. Those pathogens that breach and cross our barrier borders into the tissues below the epithelial layers will be confronted by our innate immune system's second line of defense. The second line of defense is the induced cellular responses, and this line consists of an array of innate immune cells including neutrophils, eosinophils, basophils, mast cells, monocytes, macrophages, natural killer cells, and dendritic cells. All of these cells express various receptors that gives them the ability to sense and recognize certain microbial components. So these cells act as sensor cells and play a major role in directing and initiating the various cellular immune responses, and act to amplify the strength of the innate response. Now, the specialized receptors that are expressed by our innate immune cells are called pattern recognition receptors, and these receptors can provide an initial discrimination between self and non-self. Pattern recognition receptors can detect various pathogen-associated molecular patterns that are shared by pathogens of the same type, and can also detect damage-associated molecular patterns released from the damaged cells that are under attack. These PAMPs and DAMPs are actually chemical structures or molecular motifs that shouldn't normally be present in the host, and their presence indicate either pathogen invasion or cellular damage. And what these receptors actually do is that they give our cells the ability to sense and detect these patterns, and that leads to recognition of a wide variety of pathogens and danger signals. Now, of these innate cells, we have three main sentinel cells that reside in and under the epithelium of various tissues, and these are the macrophages, the dendritic cells, and the mast cells. These resident cells, especially the resident macrophages, are the first cells to come in contact with pathogens, and they constitute a major sensory arm of the innate immune system. And these cells, along with the epithelial and endothelial cells, are the first cells to alert the immune system to the presence of pathogens. And these resident cells not only act as sentinels for invading pathogens, but also have a regulatory role throughout the course of acute inflammation. Now, upon pathogen recognition through pattern recognition receptors, what actually happens is that this recognition induces and triggers a variety of responses and defense mechanisms in these cells against the invaders, such as the activation of phagocytosis, the production of lots of mediators such as the antimicrobial substances, the degradative enzymes, and the reactive oxygen intermediates needed for microbial degradation, the production of cytokines and chemokines that convey important signals which can initiate a local inflammation and can propagate the immune response by recruiting other immune cells to the site of infection. And that's why the second line of defense is called the induced cellular response. Because triggering pattern recognition receptors on innate immune cells can activate these cells and can induce them to execute a lot of cellular responses or effector functions. And most importantly, inducing a local inflammatory response. The inflammatory response, which is initiated by our second-line innate cells, has many essential roles in combating infection. Inflammation increases the delivery of effector cells and molecules that are needed from the blood into the inflamed tissue, mainly by vasodilation, increased permeability, and increased smooth muscle contraction. The second role of inflammation in combating pathogens is to provide a barrier which kind of physically prevents the systemic spread of infection in the bloodstream, and this is mainly by the induced effect of local blood clotting. The third role of inflammation is that it changes the profile of proteins that are synthesized in the liver and secreted into the blood. And an example of such effect is seen in the acute phase response, where acute phase protein production in the liver is increased. The fourth role of inflammation is increasing the delivery of antigens from the area of infection to our lymphoid tissue, mainly by increasing the flow of lymph. We will discuss these four roles in details in a separate lecture, but I want to further address the fourth role of inflammation, which is increasing the flow of lymph and thus increasing the delivery of antigens to our secondary lymphoid organs. This process here is critical for initiating the third-line adaptive defenses. There are many cells that can carry and present antigens, but our main antigen-presenting cell is the dendritic cell. Dendritic cells, unlike macrophages and neutrophils, are not much involved in the direct killing of pathogens. Instead, they are specialized in antigen capturing, processing, and presentation. And what these dendritic cells do is that they grab antigens of the invading pathogen. They process these antigens into peptides and load them onto their surface on MHC molecules. And then they migrate through lymph to the nearby lymph nodes, where they present their cargo to naive T cells. And this process of antigen presentation is what initiates the third line of defense and is the cornerstone of adaptive immunity. The first two lines of defense are not specific for individual pathogens and are considered key elements of the innate arm of our immune response, and when breached actually contribute to the initiation and development of our third line of defense, which is specific and constitutes our adaptive arm of immunity. So the adaptive immune response is initiated when a pathogen overwhelms the innate defense mechanisms. An adaptive immune response is generated when naive T cells contact activated antigen-presenting cells in the secondary lymphoid organs. Upon this encounter, the naive T cell becomes activated and is induced to proliferate and differentiate over several days within that lymphoid tissue. And the goal of that is to generate many progeny called effector T cells possessing new activities that contribute to removal of the source of antigen, and also interact with B cells within the lymphoid tissue to provide help for the production of pathogen-specific antibodies. So the main role of the third line of defense is to provide our body with the needed effector cells and antibodies, which are specifically capable of combating the targeted pathogen.